This is the 12th lecture of our biochemistry semester, and in this lecture we'll be covering chapter 10, which covers lipids and fatty acid structure. So we'll be going through a lot of different structures of these molecules in the class. And just a kind of a breakdown of what chapter 10 entails, we'll talk about an overview of lipids, what they are, and then we'll go through three different types of lipids, how we can classify them. The first being storage lipids, probably the simplest. Then we'll talk about structural lipids. Those are the lipids found in membranes. And finally, we'll talk a little bit about signaling lipids. So they're a very diverse class of molecules, and that term lipid really just means that these molecules are not very soluble in water. And let me get my little highlighter out here. There we go. So really one of the simplest lipids that we'll talk about are fatty acids, and this is a free fatty acid. There's a carboxylic acid and then a hydrocarbon chain. Okay, very simple. You can have these kind of come together and form a triacylglycerol, which there's a glycerol here highlighted and then 3 fatty acids attached to it. That would be the major way that these fatty acids are stored in the body. And then you can also have molecules where one of these fatty acids is replaced on that glycerol with a phosphate group and then some other sort of what we call a head group. Okay, and those are structural lipids that one of the things that they do is to make membranes, right? So you have 2 fatty acid chains attached to that glycerol, then a phosphate group, and then some other sort of a head group. Okay, so those are all related to one another. You can also have other lipids that really don't look anything like that, right? So things like cholesterol has this structure, okay, and you have these 4 rings here, which is what we call the steroid nucleus, and then a long hydrophobic alkyl side chain. Okay, so these are cholesterol, and then the derivatives of that would be the steroid hormones. Okay, and then you can also have other molecules, vitamins, and this is ubiquinone. And ubiquinone you'll see again when you talk about metabolism, when you cover that material next semester. But what it has is this very long hydrophobic tail, and then on the quinone head group is used for accepting electrons and then transferring those electrons down the electron transport chain. So some biological functions of lipids. Okay, one class would be sort of the storage or physical function. Okay, storage of energy is very important. Lipids have reduced, they're very reduced compounds, and that means they have a lot of available energy. They're hydrophobic in nature, which leads to very good packing. They provide insulation from the environment. Okay? They have low thermal conductivity, high heat capacity, which means they can absorb heat. And they also do provide mechanical protection, which can absorb shock. When you're, an animal trying to move around, run around very quickly, it can provide some mechanical protection from that. And finally, some water repellent nature. Okay, they're hydrophobic, and so some organisms have these lipids or sort of waxy materials on themselves as a water repellent. The book also talks about buoyancy control and also acoustics and some marine mammals. That's really kind of not really important for our class, but you can read about that if you're interested. Okay, some more functions, and I would classify these as more structural functions or biochemical functions, right? Membrane structure. Our cell membranes are made up of lipids. And they're very important. The properties of those lipids are very important for cells. They can make cofactors for enzymes. Some lipids are cofactors for enzymes. Okay. One we'll mention, and we've sort of already seen it, coenzyme Q or ubiquinone. This is involved in the main respiratory chain, so ATP synthesis in mitochondria. Vitamin K, the book also talks about. Not really going to cover it in this class, but that's involved in blood clot formation. No, there's some other cofactors. Okay, signaling molecules is also another important sort of biochemical use of lipids. Right, we have paracrine hormones that act locally, or steroid hormones that act the whole body wide. Okay, and vitamins A and D are both hormone precursors. Okay. The book also mentions pigments and antioxidants. Those are some uses of lipids, which won't be important for our class. Okay, probably the most important is this structure of cell membranes. And as we move on to chapter 11, talking about cell membranes, that will become more important and more apparent. Right, and this is just for your information, won't be tested on this, but examples of some lipids that provide pigment in birds. Right, this lipid here, it provides a bright red color, and then this lipid here, that very similar structure. Right, we just have a slightly different derivatization on the rings at the end, and it yields a yellow color. So we can classify the lipids based on structure and function. Okay. First, lipids that don't contain fatty acids, things like cholesterol or terpenes, we can kind of move those into one group. Okay, and those would be things that are, signaling, for the most part, signaling lipids, or the cofactors, those sort of functions would the lipids without fatty acids kind of fall into. Okay. Lipids that do contain fatty acids we can also call complex lipids. They can be their own class, and that can be further categorized into storage lipids or membrane lipids. Okay. So for storage lipids, these really are the triacylglycerols. And these are cartoon figures here that show you the basic building blocks of these molecules. So a triacylglycerol, there'll be a glycerol and 3 fatty acids attached to it. Okay, we will go through these structures more in depth, so don't feel bad if you're not really getting it just yet. But this is a good overview figure. Okay, membrane lipids, you can classify these into phospholipids, glycolipids, and then there's another class that we really probably won't talk a whole lot about. We'll see an example of ether lipids, and this in particular, the archaebacterial, where this sort of a lipid is very good at surviving very harsh environments. So the membranes of archaebacteria use lipids with this sort of structure, with ether linkages. Okay. All right. So we'll go through these in some more detail throughout this lecture. Okay, so the first section of the chapter talks about fatty acids, and this is obviously a main constituent of the membrane lipids and storage lipids. So it's really important to know what a fatty acid is. Okay, fatty acids, they have, the acid name comes from the fact that they have carboxylic acid in their structure. And then they have hydrocarbon chains, and those chains can range from anywhere from 4 to 36 carbons, right? So 4 would be really, really small, 36 would be, really long. Usually you see naturally somewhere on the order of chain length 12 to 20, I think would be the most common lengths, somewhere in that range. Okay. Almost all natural fatty acids have an even number of carbons, and when you look into fatty acid metabolism, it becomes more apparent why, but the carbons are either added or taken off in series of 2 carbons. Most natural fatty acids are unbranched, so they have one chain and the chain doesn't get branched. When you say a fatty acid is saturated, that means there are no double bonds between the carbons in the chain. Okay, so that it's, in a sense, you can think of it as saturated with hydrogens. Okay. When they're unsaturated, that means there are double bonds. Okay, mono unsaturated, as the name suggests, means there's one double bond between carbons in that hydrocarbon alkyl chain. And then you can have polyunsaturated, where there's more than one double bond. So here are a couple examples of some fatty acids. And we're going to go through a little bit on nomenclature. And don't worry too much about nomenclature in this class. I'm really only expecting you to know what I would consider a specific name, and that's what I'll detail here. There's other what are called common names or classical names for these, and you won't be responsible for knowing those. Okay. So here's our fatty acid. We have a carboxylic acid, right? And that carbon in the carboxylic acid would be numbered 1. And then the chain, the alkyl chain, is shown here. Okay. And this fatty acid happens to have a double bond between carbons 9 and carbon 10. Okay, and it only has one double bond, and the total chain length here is 18. Okay, so to name this in an unambiguous way, how would we do that? Well, you have these, using the chain length, right? So 18 is our total carbons, right? So we write 18, then a semicolon, and then the number after the semicolon represents the number of double bonds. So in this case, we have one double bond in that chain. So 18 semicolon 1. That tells you the chain length's 18 carbons long. You have one double bond. And then in parentheses, we tell you where the double bond is. So parentheses delta 9 means that double bond starts at carbon 9. Okay, so that is unambiguous, tells you exactly what that structure is. You can also say the same thing in words. Okay, so this fatty acid would be cis 9 octadecanoic acid. Okay, so let's go through that. Cis means that double bond is in the cis conformation. 9 means that it starts at carbon 9. Octadec means there's 18 carbons. Enoic acid means that the ene part means there's a double bond in that structure. Oic acid means that's our fatty acid, right? This is the acid group. Okay, so both ways you should be able to find that. On an exam, I will always give you this this part, this number part, so it's unambiguous. Okay, and if I ask you for a name of something, I would really expect you to be able to give me this numbering system. All right, here's another example. So 20 semicolon 5 means that our chain length is now 20 carbons long. We have 5 double bonds, and now they're at positions 5, 8, 11, 14, and 17. Okay, the naming for this, the ECOA, is the name that you would give a hydrocarbon that's 20 carbons long. Okay, that might not be as obvious. Or ecose, I should say. So ecosa means we have a 20 carbon chain length. Penta means there's 5, and we're talking about the double bonds here because it's pentaenoic acid. Okay, and this is abbreviated as EPA. Okay, this is what they call an omega-3 fatty acid, and you've probably heard that term before. Omega-3 fatty acids are, essential for our diet because we can't make them in our body. The omega is another naming system. Okay, and what the omega numbering system is, it's starting from the end of the hydrocarbon. Okay, so we'd call this, the last carbon is omega-1, omega-2, and omega-3. So all omega-3 fatty acids, what that means is they have a double bond that is at the third from the end, the third carbon from the end. Okay. So this is omega-3. It's also an omega-6 because there's a double bond there, and an omega-9. So a lot of times you hear or see supplements that have omega-3, omega-6, and omega-9 fatty acids. That's what they're talking about. Okay, so let's do some practice drawing fatty acids. Okay, so 4 semicolon 0. Okay, that's just going to be first draw our fatty acid. Or we can have an H there, right? It doesn't really matter. Okay, this would be carbon 1. Okay, carbon 2, 3, 4. Okay, and you can draw that carbon. I guess you don't really need to draw that explicitly, right? So 1, 2, 3, 4. Okay, right, so that's pretty simple. Okay, now let's try a little bit more complex. 12 semicolon 1 delta 9. Okay, so here is our starting point again. Our carboxylic acid. This is carbon 1. Okay, 2, 3, 4, 5, 6, 7, 8, 9. Whoops. Okay, 9, there's a double bond. So now we can draw that double bond to 10, 11, 12. Okay, what you'll notice about this is also an omega-3 fatty acid. So that's omega-1, omega-2, whoops, and omega-3. Okay, finally, 24 semicolon 3, delta 9, 12, 15. There's our fatty acid at carbon 1. Okay, 2, 3, 4, 5, 6, 7, 8, 9. To 10, double bond. 11, 12. 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. Okay, so pretty, and I guess I'll just number these, 9, 12, 15. Okay, so pretty simple. Okay, how about naming fatty acids? Okay, this should also be, fairly simple. Okay, you start at the carboxylic acid. These are all going to be 1s. Okay, then we'll count over. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, whoa, 13, 14, 15, 16, 17, 18. So a total of 18 carbons in this chain. So that's 18 semicolon. How many double bonds do we have? We have 2. Where are the double bonds? Well, one starts at 9 and the other starts at 12. Okay, so that would be 18 semicolon 2 delta 9 12. Right, so what we'd call this, it would be some octadecanoic acid, but you'd have to denote that both of those are in the cis conformation. There's 2 double bonds in it. Okay, and again, I wouldn't expect you to name it that way, but if you saw the name for it, in the words, you probably could be able to pick that out. But we're going to stick to this, the numbering system, the unambiguous system. Okay. Second structure, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Okay, a total of 20. So that'll be 20 semicolon 4 double bonds. The delta numbers are 5, 8, 11, and 14. All right, and then finally, okay, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbons. So this one is 18 semicolon 0, right? Pretty simple. And the table in the book shows a little bit more about the naming systems. And can I get back to my cursor? Okay, so in this table, it'll tell you the carbon skeleton, which is really the naming system we're going to stick to in the class. Structure, don't so much care about that. Then it gives you a systematic name. Okay, these systematic names, hopefully you'd be able to recognize what the fatty acid is if I gave you one of these. But really we're going to be sticking with, if I do give you one of these names, I'd also have the carbon skeleton with it. Okay. And they're fairly easy, right? Because most of the time it'll be carbon lengths of 12 to 20. So dodeca would be 12, tetradeca, hexadeca, octadeca, and then the Archaea number, right? Because they usually occur in even numbers. Okay. What you'll also notice is the common name. And a lot of times in literature, or I know in dietary stuff, this is the name you get on a lot of these fatty acids. But those names tell you literally nothing about what the structure is. So, things like lauric acid doesn't really tell you anything. What that name comes from is, when they were first isolated, right? This came from laurel plants. So that's why they call it lauric acid. Palmitic acid, right, was isolated from palm trees. So that's where that gets that name. Okay, it doesn't really tell you anything about what the actual molecules look like. But you probably have heard some of these, like stearic acid, oleic acid, linoleic acid, right? Alpha linoleic acid. You've probably heard, these. What I will point out now, and we'll talk about this on the next few slides, is if you'll notice these melting points. Okay, the melting points for the unsaturated fatty acids are very high. Okay, they're 40s, 50s, 60s, and even up to 86 degrees Celsius. Okay, so that's pretty high. That means these are solid at room temperature. Okay, and you'll notice too the melting point is increasing with increasing chain length. When you look at unsaturated fatty acids, that melting point now is actually very low. So a 16 carbon fatty acid with one double bond is a melting point of about 1 degree Celsius. Where if that's unsaturated, 16 carbon fatty acid, its melting point is 63. So it's a huge, huge difference. Okay, and as you add more double bonds, if you look at 18 semicolon 1, 18 semicolon 2, 18 semicolon 3, that melting point goes from 13 to around 1 to minus 11 Celsius. So adding double bonds really lowers the melting point. So these are the unsaturated fatty acids tend to be liquid at room temperature. Saturated fatty acids tend to be solids. Okay. So if we look at this solubility and melting point, how they change, right? Solubility decreases as the chain length increases. So you get a longer hydrophobic chain, solubility is going to go down. Solubility is going to increase as the number of double bonds increases. Right? Melting point follows the same, inverse relationship. Okay, melting point is going to decrease as the chain length decreases. So if the chain length increases, melting point would increase. If the chain length decreases, melting point will decrease. And melting point also decreases again as the number of double bonds increases. The conformation of fatty acids, well, the saturated fatty acids, that alkyl chain is really pretty linear. Okay. When the double bond is added, when it's for an unsaturated fatty acid, that gives a kink in this structure, right? So at that double bond, most often fatty acids, unsaturated fatty acids, their double bonds will be in the cis conformation, which gives it a kink in that chain. Okay, we'll talk about trans configuration, which are trans fatty acids or trans fats. We'll talk about that here in a second. Okay. So melting point, why does the melting point get affected by double bonds? Well, saturated fatty acids pack in a pretty ordered way. Okay, they're all linear. They can pack side by side, as you see down here in this figure. Okay, and there's extensive favorable interactions there between the side chains. Okay, if you look at an unsaturated fatty acid with that the double bonds are in the cis conformation, they're not going to be able to pack very orderly, right? Those double bonds put those kinks in, and they're going to be, definitely cause this to be not ordered, not packed very closely together. Okay, so there's less favorable interactions there. All right, so it's going to take less thermal energy to disrupt that disordered packing. Okay, and that's why their melting points are so much lower. It doesn't take as much heat energy to disrupt that and to make them into liquids. Trans fats or trans fatty acids, they're really not natural, naturally formed in the body. They're formed by partial dehydrogenation of unsaturated fatty acids, and they're done to increase shelf life or stability of certain food products at higher temperatures, right? Especially for deep frying, okay? So that's kind of where trans fats originated in the diet, okay? Probably not a whole lot left anymore because of how it became such a hot topic in nutrition to kind of eliminate these. So I don't know how much of them are used anymore, but that's sort of what historically what they were used for. Okay. A trans double bond allows a given fatty acid to adopt an extended conformation. So instead of having a kink at the double bond, they're actually come back to being very, very linear. And trans fatty acids can pack much more regularly, even than a particular saturated fatty acid. And so they have higher melting points than their cis forms. Okay. So eating these trans fats increases the risk of cardiovascular disease because they they have lower melting points, higher melting points, and they become solid in your body and in your arteries. Okay. So just avoid frying in these sort of vegetable, partially hydrogenated vegetable oils, right? Okay. So if we look at a picture of these, right, a saturated fatty acid, very linear, right? Source where you would get those, you'd find them typically in animal fats, okay? They're solids, room temperature. A lot of animals that use fats as a way of insulation would use more saturated fatty acids. Okay, unsaturated fatty acids where we have a cis conformation at the double bond. These tend to be more plant type oils, things like olive oil. Okay, they're liquids at room temperature. And then trans fats come again chemically processed oils, where you would take something like the olive oil or some sort of a vegetable oil and do a partially hydrogenated version of that, and it would change these into trans fats. And again, that structure gets very extended, linear, and so that provides very ordered packing, okay, lowering the melting point. Okay. Right, so that's fatty acids in sort of an overview of those. And the fatty acids are building blocks for this storage of fat in the body, and those are in the form of triacylglycerols. Okay. So the majority of fatty acids in biological systems are found in the form of triacylglycerols. Okay, solid ones are called fats, and liquid ones are called oils. Okay, primary storage form of lipids in the body, in the form of body fat. Okay, in our bodies. Less soluble in water than fatty acids by themselves because they lack that charged carboxylate group, and we'll see the structure here in a second. But that where we normally have a negative charge on the carboxylate group, it is now attached to a glycerol. Okay. These are less dense than water, and that's why fats and oils float on water. So our structure for triacylglycerol, okay, and we'll highlight the structure of glycerol here in red. We have what glycerol is it's a 3-carbon chain, and there's an alcohol group at each carbon. So on those alcohol groups, those were combining with our fatty acids to make this linkage. Okay, so here we have we number these 1, 2, 3, right? 1 and 2, 1 and 3 carbons 1 and 3 are really equivalent here. because if you just rotate that molecule around, 1 and 3, you can't tell them apart. But carbon 2, you should always be able to tell that it's the one in the middle. Okay, so here we have our triacylglycerol. At carbon 1 here, it's this stearic acid. And this is linoleic acid and palmityl acid, right? So that's what it's 1, 2, and 3, these fatty acids there. Right, so again, at this class, I would give you that numbering system to name those. And then finally, it would be glycerol, because they're all attached to glycerol. Yeah. Yeah. The advantage of fats for energy, they're actually really, really good to use as an energy source. And why they're so good is that they carry much more energy per carbon because they're fully reduced, or most of the carbons in them are fully reduced. And what that means is they have, you can think of it in terms of hydrogens. They have the most hydrogens they can have on those carbons. Okay. Things with double bonds or things with oxygens attached to them would be less reduced or more oxidized, and therefore give less potential energy. When you cover metabolism, you'll probably talk about this again, and it might make a little bit more sense after going through metabolism. Okay. The other important advantage of fats over polysaccharides, which are long chains of sugars, fatty acids carry less water. That's because they're nonpolar. Sugars, polysaccharides, are polar, so they're hydrated. Water molecules go with them. So because fats don't carry those water molecules, they're not as heavy. And if you're trying to store a lot of energy in your body and you don't want to weigh yourself down, right? So fats are actually, really good at storing energy without also having to weigh the animal down with a lot of water weight. Okay. Glucose and glycogen, those are sugars, are good for short-term energy needs, right? Quick delivery. Fats are really for longer-term energy storage, up to months of energy needs. So you can store fats, you can store them for a very long time, and then they give a slower delivery of energy as well. Right, so you could use sugars when you are intaking food in a fed state, but then in fasted states you sort of switch to the fat energy usage. Right. This is just a picture of some fat cells in the body, and you can see that they're really clear because they're just, all they do is store fat. They really don't have a lot else going on in them. The book talks about waxes too, and we really won't cover waxes in this class. You won't really be responsible for them, but if you're interested, what's the difference between a fatty acid and a wax? Well, waxes tend to have a fatty acid component to them, and then where that acid group is, they have another really long carbon chain attached there. So this one, right, there's 28 carbons here in the middle here, and 29, 30, so a total of 30 carbons here attached there. So that makes them, really, really water insoluble, okay, and very high melting points. And things like that, this triacontanol is also used. So that was a sort of an added to a fatty acid to make a wax, but bees also use fatty acids derivatized with that to make their hives. So out of what's known as beeswax. Okay, right. So we've already kind of covered this table, so I don't really think I need to say much else about this. But just another place for you to see this table, have this reference. Okay, moving on to the second section. Okay, in the second section, we talked, first section was about storage lipids, and those are the triacylglycerols. The second section deals with structural lipids. And these structural lipids, what we mean by structural is that they're lipids that can form membranes. Okay. Structural lipids, the unique thing about them is that they do have a polar component. Okay, so they contain polar, what are called head groups, and then their nonpolar part, the tails, are usually fatty acids or some sort of fatty acid type molecule as their tail group. Okay. These can be very diverse, and that diversification can come from modifying a different backbone, changing what the fatty acid groups are, and modifying the head groups. Okay. Properties of the head groups really determine the surface property of the membrane, as we'll see when we talk about membrane structure. The fatty acid unpolar, nonpolar parts are going to be in the middle of the membrane. The polar head groups are what stick out on the surface of the membrane. So changing that head group out is going to change the surface property of a membrane. Right. And different organisms have different membrane lipid head group compositions, and different tissues have different membrane lipid head group compositions. And when we cover chapter 11, we'll get into some of those differences, especially the tissue difference and the organism difference. We'll talk about both of those. The most common membrane lipid that we'll focus on are those that are known as Glycerophospholipids. Okay, these are the primary constituents of cell membranes. They have two fatty acids that have these ester linkages with carbon 1 and carbon 2 hydroxyl groups of glycerol. Okay, and not just glycerol, but glycerol 3-phosphate. So you can think of it as, glycerol, fatty acids attached to carbon 1 and carbon 2 via ester linkages on their hydroxyls. And then at carbon 3, you have a phosphate group. So here, this is our backbone of glycerol phospholipids. It's glycerol 3-phosphate. The head group is charged at physiological pH. So the phosphate group here is going to be charged, and the head groups that we add, the other head groups we add at the phosphate on carbon 3 to this will also be charged, at least most of them. Okay. So if we look at the structure in more detail here, in red, this is our glycerol. Okay. We have fatty acids attached at carbon 1 and carbon 2, right? And they're attached by this ester linkage. So we have what normally would be the carboxylic acid. It's actually attached the O, where it would normally be an OH or an O minus, is now attached to both the fatty acid chain and the carbon of glycerol. So in this case, it's carbon 1. Okay, so that's the ester linkage. Right, so we have fatty acids at carbon 1 and carbon 2. And then at carbon 3, we have the phosphate group, and then a head group is attached to that phosphate. Okay. Unsaturated fatty acids are commonly found connected to carbon 2. So if I give you, if you ever have to draw a fatty acid, and you're given an unsaturated fatty acid and a saturated fatty acid, the unsaturated fatty acid is going to be at carbon 2. Okay. The head groups are we'll see different head groups here, but this is one way to diversify glycerol phospholipids. One way is, you can have different fatty acid chains here, and another way is you can have different head groups, and we'll talk about the head groups. Right, so the 5 that you really need to know are the first 5 in this table. Okay, so the head group name, for the first one, it really isn't anything. Okay, that glycerol phospholipid just has the phosphate group, and that's all. Then there's ethanolamine, choline, serine, and glycerol. Okay, so ethanolamine, what is that? It's 2 carbons, and it would normally be an OH right here for ethanol, but that O would be on the phosphate group too. It'd form that linkage with the phosphate. So now it's not shown here. Okay. So that would be ethanol, 2 carbon alcohol, plus the amine group. So ethanolamine. Choline, again, there's 2 carbons. Typically, it would normally have an OH group there, but again, that's not shown. So we have 2 carbons, and now instead of an amine group, we have the nitrogen, but instead of hydrogens on it, there are 3-methyl groups. Okay, so that's choline. Important to point out here, this is not showing. There's a typo here. There should be a plus charge on that. Okay, so just be aware of that. There should be a plus charge there. So ethanolamine and choline are both positively charged head groups. Okay, serine is just the amino acid serine. Okay, so there's the acid part, there's the amino part, and then here there'd be an OH, the side chain OH group is, again, not shown because that's going to be bonded to the phosphate. Okay, and finally glycerol, which again is just a 3-carbon chain, and each carbon has an OH group on it. Okay, but the one that attaches to the phosphate, we don't show the OH. Okay, so those are the ones I'd expect you to know. Also expect that you know their charges. So remember, ethanolamine is a positive charge, choline is a positive charge. Serine is net neutral, but there is a negative charge and a positive charge in it. Okay, and glycerol is a neutral charge. Okay, why does that matter? Well, it matters because in there, when you look at the glycerol phospholipid, remember that the the phosphate group is going to have a negative charge to it. So that negative charge plus whatever the charge of the head group is will tell you the net charge of that glycerol phospholipid. Right, so if there's just a phosphate group on that phospholipid, its net charge is going to be -2. Okay, the ethanolamine or the choline, they're going to have positive charges. They'll cancel out with the -1 charge of the phosphate group. They're going to have a net of 0. And these serine and glycerol both are neutral, so the only charge that you'll see is from the -1 from the phosphate group. So those both have a -1 charge. Okay. And when we put these head groups on our glycerol phospholipids, what do the names become? Okay, so if it's just a phosphate group, what we call that is phosphatidic acid. When we have ethanolamine as our head group, we call this phosphadidyl ethanolamine. So the phosphadidyl base is really telling you it's a glycerol phospholipid. And the next part is the head group. Okay, choline would be phosphatidylcholine, serine would be phosphatidylserine, and glycerol is phosphatidylglycerol, right? Pretty easy to remember those. Okay, there's some other ones here that are much more difficult, right? And they're not really just used for membranes. They're also can be used for things like signaling We'll talk about cardiolipin a little bit later and see its total structure, because this is a little bit more confusing showing this. Okay, so first, the one we'll really talk about in depth is phosphatidylcholine. Okay, this is the major component of eukaryotic cell membranes, and in fact, most prokaryotes can't even synthesize this lipid. Okay, so this really, you can think of it as a eukaryotic only membrane lipid. Okay. And again, it has a glycerol group, 2 fatty acids attached via ester linkages, and then at the other end we have a phosphate attached to our 2 carbon and our guanylyl group with the nitrogen with 3 methyl groups around it. Yeah, I think that's all I wanted to say about that one. But you'll notice here, this is a better picture. The plus charge on the nitrogen and the minus charge on our phosphate will cancel out, will lead to a neutral surface charge. So if our membrane only has phosphatidylcholine, it'll have a neutral surface charge. Okay, there's still charges there, but they cancel each other out. Okay, so those are, if we have this sort of glycerol phospholipid backbone, and we can change out these head groups, right? So if this was phosphatidylserine instead of choline here, we'd put on serine. Okay, so that really doesn't change any other part of this molecule. They're still connected here to fatty acid chains via these ester linkages. There's some other different looking membranes that have what are known as ether linkages. Okay, and so we'll go through 3 of them that will be important to recognize. I wouldn't expect that you'd be able to draw these from scratch, but just being able to recognize them. And how you can recognize them is highlighted here in red, right? That's an ether linkage. It's missing the double bond, the carbonyl, the carbon double bonded to oxygen here. Okay. So the first one we'll see is what's known as plasmalogen. Okay, and this is it's a derivative of ethanolamine, phosphatidylethynolamine. So there's on our phosphate group, there's the ethanolamine head group. Okay, but now instead of having a fatty acid here at carbon 1, that oxygen was removed, and it's now an ether linkage. Okay. This is common. We know it's common in invertebrate heart tissue, but we really don't know the function of it. One thing that we do know about it is that because this is an ether linkage and not an ester, it's resistant to cleavage by enzymes that cut off that fatty acid group, right? So that probably has something to do with its function. Okay, so that's plasmalogen. Okay, how would you know that's plasmalogen? Well, it has the ethanolamine head group, and there's this ether linkage at carbon 1 with a double bond close by. Okay, so that's how you'd be able to pick that one out. Okay, the second ether lipid is something known as the platelet activating factor. Okay, what's different about this one? Well, it has choline here as the head group. Okay, again at carbon 1 there's an ether linked, but this time there's no double bond, so it's an alkane. And then at carbon 2, now we have an acetyl group. So it's not considered a fatty acid because it's only 2 carbons long, but it's really a carboxylic acid with 1 extra carbon, so an acetyl group. Okay, so that'd be how to easily pick that one out. Right. And this is the first signaling lipid that was actually identified, and what it does is it stimulates aggregation of blood platelets, and that plays a role in mediating inflammation. Okay, finally, the third ether-linked lipid that we'll talk about are Archaea membrane lipids. Right. So if we look at these Archaea, in particular this species, where do they live? They live in very, very extreme environments, like volcanic hot springs, where you have high salt levels, you have high temperatures, and also very acidic environments. So normal plasma membrane lipids would be, very easily degraded. The membrane wouldn't have a very good consistency or integrity because at temperatures that high, it would really be very, in a sense, it'd just be a liquid, so there wouldn't be any real membrane. So these structures, and if you look at the structure, they have a glycerol here in red, and then at 2 carbons, they're linked to what you would normally think of as fatty acid chains, but these are a little bit different. You'll notice that they have these branch points here, so they have these extra methyl groups sticking out. Okay, and you'll also notice that isn't an acid or an ester linkage here; it's an ether linkage. Okay. And then we have our glycerol phosphate here would be, I guess, considered our head group. So phosphoglycerol attached to another glycerol, and then we have these methylated alkyl chains. But on the other end, right, it doesn't just end. There's now another glycerol, and then we have, some sugars. So these are alpha glucose and galactose. Okay, again, I wouldn't expect you to draw this at all, but if you saw a picture of it, I'd hope you'd be able to say, oh, there's, what is this? Well, there's ether linkages on both sides. It must be this Archaea membrane. Okay, and what this does, why is it better for these organisms? Well, the ether linkages are resistant to hydrolysis, so that helps at low pH. Okay, these branch chain methyl groups in the hydrocarbon, they're resistant to oxidation, so there's no oxidation that would occur at say, a double bond there. So that's better at high temperatures. And the hydrophilic groups on both sides, what that gives you, that gives you the ability to use this in a monolayer instead of a bilayer. Most membranes, as we'll see in chapter 11, are bilayers, so you need one layer on top, one layer on bottom, right? This would only need one layer total, right? So the inside of the cell would be on this side, and the outside could be, over here. And again, that'd be much more resistant to higher temperatures. Okay. Another, I guess you would say, a little bit different membrane lipid that we can have, opposed to a glycerol phospholipid, would be something known as a sphingolipid. Okay, on these, the difference between a glycerol phospholipid and a sphingolipid is the backbone. Okay, the backbone of sphingolipids is not glycerol. It's something called sphingosine, and we'll see that structure here in a second. Okay, fatty acid is joined to sphingosine not by a ester linkage, but by an amide linkage. Polar head group is still connected to sphingosine by either a glycosidic, so a sugar, or a phosphodiester linkage. Okay, and a sugar-containing glycosphingolipids are found largely on the outer face of plasma membranes, where that sugar can actually be used as a sort of a recognition device. Okay, so a picture of a sphingolipid is shown here. So sphingosine is highlighted in red, and what you'll notice is, you still have this 3-carbon base here that sort of looks like a glycerol, but now instead of a fatty acid chain being attached to the hydroxyl here, we have the hydrocarbon chain just continuing at that carbon, what would be carbon 3 in this case. Okay, and at carbon 1, we'd have that hydroxyl, which is then connected to the head group, which you can have different head groups, again shown down here. These head groups, not as important to know for sphingolipids. I wouldn't really ask you to draw a sphingolipid. You should be able to recognize it though. Okay, so this OH group here, that's a big clue. The other big clue is now at position 2, where we have our fatty acid attaching. Right, this is an amide. Not there's no O there, so it's not an ester linkage. It's an amide linkage. So there's a nitrogen group. So it's the sphingolipid. Those are the lipids that have this nitrogen group there. Okay, an important sphingolipid is sphingomyelin. Sphingomyelin, we have ceramide, which is the ceramide is sphingosine plus the amide linked fatty acid, which would be, this red part and the yellow part, that would be ceramide. Okay, plus phosphocholine attached to the alcohol. Okay, so we have, this would be our ceramide, right, sphingosine, and our fatty acid attached down here to the nitrogen, and then that head group on this would be a phosphocholine. Okay. Sphingomyelin is abundant in the myelin sheath that surrounds nerve cells. So right, that's where it gets its name sphingo, meaning it's a sphingolipid, and myelin because it comes from the myelin sheath. Right, and we think it's important as sort of an insulation to those nerves. Okay, it's very structurally similar to phosphatidylcholine. Okay, they both have phosphocholine head groups. Right, the differences if you saw these pointing out, the obvious difference is that this fatty acid in sphingomyelin is saturated, and in phosphatidylcholine here it's not. But these fatty acids can change a little bit depending on cellular conditions. Okay, so really the focus you should have is this amide linkage versus the ester linkage. Okay, and then here you have another ester linkage, where in sphingomyelin you have an alcohol group because this is sphingosine. Okay. We can have sugars attached to glycerol phospholipids and sphingolipids as well. Okay, and those sugar groups, one of the uses of these sugars is the blood groups. Okay, the blood groups are going to be determined by the types and order of sugars that are located on the head groups in these glycosphingolipids. So anytime you see that term glyco, either phospholipid or glycosphingolipid, glyco means there is sugars attached. Or even sometimes you'll see the term glycoproteins. Right, glyco is just another way of saying sugar. Right. The structure of the sugar is going to be determined by expression of specific glycosyl transferases, and that's where the different the blood groups come from. And you don't have to know these or be able to recognize these in this class. That's a little bit beyond the scope of it. We haven't talked about sugar structure yet, so really all we can do right now is show these sort of cartoon pictures of the different sugars involved. Okay, but I wouldn't even expect you to know the pattern or anything, be able to distinguish O from A or A and B, so this is really more for your information. Okay, and the having defects in turnover of membrane lipids or the the addition or subtraction of some of these sugar groups to these glycolipids and membranes leads to a number of diseases. And maybe you've heard of some of these, something like Tay-Sachs disease or Niemann-Pick disease, right? Niemann-Pick disease is the enzyme here, sphingomyelinase, takes sphingomyelin and breaks off a phosphocholine from it, and then the ceramide group can be recycled, right? If there's a defect in that enzyme, that leads to Niemann-Pick disease, which is a neurological disease. Right, so just in kind of a overview of what we've talked about with structural lipids, right? We've talked about phospholipids, and the two classes of those were glycerophospholipids, where you have glycerol as your backbone. Okay, you have glycerol as the backbone, two fatty acids attached, phosphate group, and then a head group. Okay, sphingolipids, you'd have sphingosine as your backbone, one fatty acid attached, phosphate, and then a choline. You can also have glycolipids. You can have glycosphingolipids, okay, where again sphingosine's your background, backbone, one fatty acid, and then some sort of a sugar group attached on your head group. A glycophospholipid, or also referred to as galactolipids. Again, you have glycerol as your backbone, two fatty acids, and then some sort of a sugar group attached as your head group. Okay. Primarily going forward, we're really going to be focusing on the phospholipids and not the glycolipids. Okay, moving on to section 3. Okay, these are signaling lipids, or signaling functions of lipids. And the first one we'll talk about is sterols. Okay, sterols, things like cholesterol would be really the one that we start with. That's the basis of these. They have this, what's known as the steroid nucleus. Okay, a steroid nucleus are these 4 rings, right? 4 connected rings, A, B, C, and D. Okay, hydroxyl head group, that would be, you can consider that their polar part, polar head group. And then they have various non-polar side chains here, right? So methyl group at 18 and positions 18 and 19, or carbons 18 and 19, I should say. Okay, this steroid nucleus, because of the rings, how they're structured, it's almost planar. Okay, so that's cholesterol, and cholesterol has also this longer alkyl side chain. Right. Their cholesterol is important in our cell membranes as well. So we think of sterols as being precursors to hormones, which are signaling molecules, but cholesterol also does play a role structurally in membranes. Okay, and it's present in membranes of most eukaryotic cells. What cholesterol does when it's in the membrane is it modulates the fluidity and the permeability of that membrane. Okay, it can thicken the plasma membrane, and most bacteria lack sterols. They can't produce sterols. Right. Mammals obtain cholesterol either from food, or they can synthesize their own in the liver. Okay, cholesterol moves around in the body when it, not freely generally, but when it's bound to proteins. And there's either low density lipoproteins or high density lipoproteins. Low density lipoproteins, or LDL, are considered the bad ones, because these tend to deposit in arteries and lead to artery clogs. So if you ever go to the doctor and they give you, like a cholesterol test with HDL and LDL levels, that's what they're referring to, and the LDL is known as the bad cholesterol. Okay. Right. Many hormones are derivatives of sterols and also in cholesterol specifically. Okay. Steroid hormones that are derived from sterols, they're just oxidized derivatives of sterols, right? And they have that sterol nucleus, the steroid nucleus, the 4 rings, but they lack that alkyl chain found in cholesterol. So because of that, they're a little bit more soluble and more able to, they've been oxidized and they lack that chain, the hydrocarbon chain, so they're more soluble and they can flow freer through the body. And they're obviously, because of that, more polar than cholesterol. The hormones, steroid hormones, are synthesized from cholesterol in different parts of the body, gonads, adrenal glands. They're carried throughout the body in the bloodstream, usually attached to carrier protein still, so they're not, freely flowing around. Many steroid hormones are the male and female sex hormones. Right, and so here's an example of some of them, what they would look like. You probably heard of a lot of these, actually, like testosterone. I'm sure everyone's heard of that, the male sex hormone. And it has a fairly simple structure. Cortisol, that's been known as a stress hormone, but it's also involved in regulating the body clock, internal clock, right? So it has a slightly more derivatized than testosterone. And then some of these other ones maybe you might not have heard of. Prednisone, you maybe heard of that one. That you can actually take prednisone for certain disorders or to combat, very bad inflammation or I know for instance, if you have a really bad gout flare, sometimes they'll prescribe prednisone. Okay. Don't expect that you would be able to, draw these or even recognize them. They're all very, very similar. That's kind of what we're pointing out here, that they're 4 rings. That 4-ring nucleus, steroid nucleus, is seen in all of these. They're just, the double bonds are slightly different. There's oxygens at different points, right? If I showed you a picture of one of these on a matching question, I'd hope that you'd be able to say, oh, that's going to be a hormone, right? Or if I showed you cholesterol, you'd be able to say, oh, that's cholesterol, because they do look wildly different than the other lipids that we've seen. Okay. All right, some other biologically active lipids. Okay, they're present in much smaller amounts than storage or structural lipids, right? You don't need as much of them, but they still play pretty important roles as signaling molecules. And some of the examples are the lipid-soluble vitamins. So vitamins A, D, E, and K are all lipid-soluble. Okay, and they have they're what you would say biologically active. They play a role either in signaling or in antioxidant. Okay. So the book gives this example of something known as arachidonic acid. Okay, arachidonic acid, or arachidonate, is shown here. If you count up those carbons, it's a fatty acid that's 20 carbons long, and there's 4 double bonds, 5, 8, 11, and 14. Okay, so that's arachidonate. Okay, and it actually has a few different roles in different pathways. In one pathway, it can signal and kind of get converted into what's known as prostaglandin, and that would signal inflammation in your body. Okay, that's a big signal of inflammation and fever. Okay, it can also be converted into thromboxane, and that signals to get to the formation of blood clots. And this leukotriene is the other one, and that's smooth muscle contraction in the lung. So we won't really talk about that one. These other ones, prostaglandin and thromboxane, those are the ones that I would expect you to kind of remember, okay, right? Prostaglandin is for fever and inflammation. Thromboxane is blood clotting, right? And so what a very widely used class of drugs, the NSAIDs, that are non-steroidal anti-inflammatory drugs, those block these both these pathways, right? It blocks the formation of prostaglandin and thromboxane. So that's how those drugs work against inflammation and fever, and they're anticoagulants because they block that formation of thromboxane. Okay. Okay, vitamin D is another biologically active lipid. Okay, and this isn't vitamin D. It's cholesterol, or more precisely 7-dehydrocholesterol. Right. So this molecule, when exposed in your skin to UV light, it turns into vitamin D3. Okay. So this is vitamin D3, and then vitamin D3 will turn into in the liver and the kidney into what's known as calcitriol, which is the biologically active form. Okay, so it's important. Everyone kind of, I think, knows vitamin D comes from getting UV light, right? But what's interesting is it's not just having UV light. You also have to have cholesterol. So cholesterol is very important part of the diet, and it got sort of a really bad rap there for a while. But it is important, and I think in general our diets are probably fairly low in cholesterol now. And I actually was tested as having a vitamin D deficiency one summer, because in this summer, like, I was riding my bike all the time, like 10 hours a week out in the sun, but we're using more sunscreen, right? And our diets are probably fairly low in cholesterol, so we don't have the precursor cholesterol in very high levels necessarily. And by using sunscreen, maybe we're not giving quite as much UV light. So there can be lots of people that have vitamin D deficiencies and probably don't really know it. And vitamin D has been more important now in the news, because they're showing that vitamin D is actually the vitamin D levels can kind of correlate with who's getting bad symptoms from having COVID. Having a lack of vitamin D can actually hurt your immune system and the ability to fight off the COVID infection. So just something to kind of be aware of. Right, and the last biologically active lipid we'll look at is vitamin A, also known as retinol. Okay, this is involved in visual pigment. Okay. And really where you get this in your diet is from eating vegetables that have beta carotene. Okay, and beta carotene looks something like this, and it has a point of cleavage in that molecule where it kind of splits in two. Okay, so you'll break it down when you ingest beta carotene. It breaks in two, and then it becomes what's known as vitamin A1 or retinol. Okay, this molecule can then be used. It's converted a little bit here. There's some changes. The most important thing to note is that there's now a cis double bond in it. Instead of all trans double bonds, there's one cis at position 11. Okay, and so this is what we call 11 cis retinal. And this is the visual pigment. So this is what is used in your eye cells to pick up visible light. Okay, when a photon of visible light hits this cis double bond is converted to a trans double bond, right? So it goes from cis retinal to trans retinal, and that changing that conformation there is enough to trigger a nerve signal to your brain, and then your brain picks that up as sight. Okay, so this is, and we'll see this again when we talk about signaling pathways. We'll see this molecule again. Okay, some other biologically active vitamins, vitamin E, vitamin K, and some other lipid quinones are shown here, right? So this is vitamin E, typically thought of as an antioxidant in your body. Vitamin K for blood clotting. Ubiquinone that we've seen, electron transporter. Okay, they all have these long isoprene tails, but their head groups are sort of what their function comes from. Right, so next lecture we'll be covering chapter 11, and that will really be dealing with membranes. Okay, the composition of membranes, how the lipids are dynamic in the membrane, and then transport across the membranes. Okay, and just a reminder, we have our second in-class activity on October 15th, talking about mitochondrial protein transport. So you want to make sure to go to the class meeting for that.